All right, you all heard it. I just hit the record button for legal purposes, to say it out loud. Yeah, so I'm apologize to you, Dr. Rinkevich. I don't have a huge intro. I didn't ask for your CV, but you work for the Ag Research Service, USDA. You are in Baton Rouge, correct?
Yeah,
okay,
yeah,
awesome.
That's right. That's right. Yeah. Well,
yeah, go ahead. Take it away.
Yeah, I was gonna say I live in Baton Rouge now, but I'm originally from Pennsylvania, and I actually went to Millersville for my undergraduate. And when I was coming up to think about talking today, I found this beauty in my closet. This shirt's like 25 years old. I got it when I was undergrad at Millersville. So it's always cool to have a connection to Lancaster, with, not only through university, but also through now with beekeeping. So thank you very much for inviting me to come speak at this meeting here. This is highly enjoyable for me. So I'm going to talk to you today about this Varroa sensitive hygienic bees that we've raised here at the lab. So I work at the honeybee breeding genetics and physiology lab in Baton Rouge. And as you can imagine, that involves a lot of breeding genetic experiments and things like that. But it also really focuses not just on the honeybee, but a lot of what we do is focus on Varroa. So one of the other things I work on is amitraz resistance in Varroa. And then I'm also being a steward of these VSH or POL-Line bees that we're going to talk about today. So bees are an agricultural entity, right? They're considered livestock now. They have been for a few years. And with that in mind, we have to think about how they are amenable to being agriculturally important. So like I said, we work in genetics and physiology and breeding. And so this selective breeding is actually all around us in agriculture. And I'm sure you've heard of some of these examples that corn has been selectively bred from this little piddly looking thing here called teosinte to the large corn that we traditionally enjoy today. So after many years of selection, getting those kernels bigger and the cobs bigger as well, we end up with this modern corn. Same
Dr.
thing.
Rinkevich, one sec, I apologize. I have to make you a host.
Oh.
So you can.
Oh my God. I totally forgot to share my screen. So you're probably.
Well, and I forgot to make you the host. You wouldn't have been able to. Go ahead.
Okay. I got too excited. I got too excited.
All the time. You know, like you start talking and you can't. Nobody can see what you're doing.
Yeah. You're probably like. All right. Now, could you see something?
We got it. Yeah.
All right. Good. So good. Thank you for stepping in about that. Yeah. So, like I was saying, is that corn started off as this little thing here called teosinte, and then after many years of selection, it gets to this big corn that we enjoy today. Same thing with like cabbage, broccoli, cauliflower. This kind of weird looking brassicaceous plant, you select for different criteria and you get things like kale, kohlrabi, Brussels sprouts, cabbage. It's all the same species of plant, just selectively bred for these different features. So we could do that with plants, but we could also do it with cattle and things like that. So they could be raised for things like milk quality, or in the case of like the Jersey breeds, they are mostly bred for their high butterfat content, so make good butter. And then also too, there, as I'm sure a lot of us know, that there's different breeds of cattle that are raised for their beef. So all the same ones, but just with different criteria for what's valuable to those ranchers. Bees, believe it or not, are actually no different. They can be selectively bred for different traits. And they could be as obvious as ones of like high honey production. You can see that here. These are frames that are just loaded with honey and from the result of a selective breeding program. The color of bees, you could easily breed for that. You just keep breeding them based on which color you like. Marla Spivak from North Minnesota was able to breed bees that do this hygienic behavior. So they removed the diseased and dying brood. This is a highly hygienic colony. And that's, again, something that has a genetic component. You can breed for it. Another desirable characteristic is large honeybee populations, which is exactly what everybody wants. You want large populations. So that's been passively selected for over thousands of years of honeybees that have been maintained by humans. And then finally, we could also select for things like large pollen sources. Like I said, honeybees are no different than any other livestock, and it actually forms the basis of our lab here in Baton Rouge. Like I mentioned, we are the Honeybee Breeding Genetics and Physiology Lab. All of us at the lab work on bees, but we have different interests. So my interests are in toxicology. So I'm trying to breed bees that are resistant to pesticides. We're also trying to breed bees that are resistant to viruses. We're also trying to breed bees that actually have better feed conversion. So for example, in the poultry industry, that's like the benchmark of feed that makes you a successful chicken farmer is feed conversion rates. But no one's ever done this for bees. So we're trying to breed bees for those feed conversion rates. So among many, many other things that take many years. So that's a little bit about what we do at the lab. We are in service of the American honeybee keeping industry. So we work alongside a lot of the commercial beekeepers. And when you ask commercial beekeepers, what's really affecting the bees, they almost always say it's this one thing, Varroa. Everybody hates Varroa. Because if you keep bees, you're going to have Varroa. And you don't have Varroa because you're a bad beekeeper. You have Varroa because you have bees, you know, just like a dog could get fleas or a cat could get tick on you. It's the same thing. They're just a parasite of one of our favorite insects. And so what this graph here shows is that when they asked beekeepers of different sizes, so they looked at backyard beekeepers, so this is like 5 or less colonies. Sidelines are about 5 to 50 colonies. And the black bar here shows the commercial beekeepers, so these are the large scale people. They asked them what's affecting their bees. So they had a variety of responses down here, as you could see. And the commercial beekeepers say that Varroa is the most important factor, and you can see it here by this large bar. So that's kind of obvious. But actually, I think this bar should be higher because when you look at things like weak colonies, well, what is a weak colony? Don't know or other, those are actually probably something to do with Varroa. So Varroa is definitely the major cause of colony losses, and I think it's actually underestimated. So not only does the feeding of Varroa damage honeybees, they really do a number on our bees by transmitting a lot of viruses. So the foreign wing virus, which I'm sure you'll see in the fall with high Varroa populations, but then also chronic bee paralysis virus. This is kind of an interesting one because they look shiny and like they look wet and oily. They don't have any hairs on them. They kind of crawl around really slowly. So these are the two major classes of viruses that are transmitted by Varroa. So because of that, a lot of beekeepers know to control Varroa, you could use chemical control. And these are just a few examples of some of the currently registered products to control Varroa. When used appropriately, according to label conditions, make sure you're using it according to label conditions, these materials do work. However, in some cases though, we're starting to get resistance to some of these products. And like I mentioned before, I really work on amitraz resistance in Varroa. So across the United States, the last 3 years, I've been doing this big project where we look at the amitraz resistance in Varroa. And what this graph here shows is on this axis, these are just different beekeeping operations, you can see here. And this is what's called apivar efficacy. This is a measurement of amitraz resistance. If the bar is really high, they're susceptible to amitraz. If the bar is really low, that means they're resistant to amitraz. And what we have here in red is the threshold for resistance at the colony level. And what we have here is these colonies that are shown in red, but with the stripe marks on them, that is the apivar efficacy in apiaries in which the beekeepers say amitraz doesn't work. So you can see here that these are correlated, these control failures at the colony level are falling below the 70% threshold. When we look at the proportion, about 24% of all the apiaries that we sampled over the last 3 years are resistant to, the Varroa are resistant to amitraz. So this is something that we have to pay attention to because we can't be relying only on chemical control measures because resistance can evolve, making those products less effective. But the interesting thing is that honeybees can actually survive Varroa infestation. So Varroa is a worldwide phenomenon. It's found everywhere except Australia and a few other outlying places. They really do a number on feral bee colonies. So for example, in California, in the early 90s, when Varroa got there, it eliminated about 75% of the feral honeybee colonies. But because honeybees are able to adapt to these pests, there have been instances where they can survive without any kind of chemical intervention. So this map here shows the location of a number of honeybee populations that could survive Varroa without any chemical miticides. So they have some kind of resistance to them, some sort of behavior, some sort of characteristic that makes them tolerate Varroa. And so when we open up these colonies, actually take a look at how do they keep Varroa populations low, there's a whole bunch of different ways that can happen. So if we're looking at the different traits or how they get there, here's just a couple examples of them. So infested brood removal, and that's what you're seeing here. You see that Varroa on this pupa? These bees are actively removing that pupa and discarding it. That's a hygienic behavior. You could think of it similarly if that brood was infested with chalk brood or some sort of a bacterial pathogen, they will remove that brood. That's one of the important parts of it. Another way that they could do this is with suppression of mite reproduction, or SMR. What that behavior means that the Varroa, typically when they get into the honeybee cell, they'll lay a couple of eggs, usually one that's the male and then the 3 other daughters. In bees that have this suppression of mite reproduction, those foundress Varroa never lay any eggs for some reason. So, and it's still unclear how that actually happens. There's another program called social apoptosis, where the larvae, or the pupa, I'm sorry, actually kill themselves in order to prevent the Varroa from reproducing. And that's actually how the eastern honeybee, Apis cerana, deals with Varroa. There's other populations that have this grooming behavior where they can knock the mites off of them. And then there's another group typically called the ankle biters, where they'll actually chew the legs off of Varroa so they can't get around. And that's what you see here. This is a leg, and this is a Varroa that had its legs chewed off. But some of those populations that I showed on the map have some qualities that allow them to survive Varroa, but aren't really good for beekeeping, such as small colony size, and they swarm a lot. So they might swarm 3 or 4 times a year. And then also to small cell size, so the size of the cells that the brood are laid in. So we know that honeybees can survive Varroa, but how can we breed for that Varroa resistance? So this whole project started about close to 25 years ago now at the lab. So John Harbaugh and Jeff Harris are lab alumni who initiated this project to select for Varroa resistance based off of behavior. And the first step that they wanted to do is they wanted to find out what characteristics of low Varroa populations were heritable, meaning did they have a genetic component? Because with Varroa population, it could be genetics or it could be environment. Because the higher the heritability, the better that is to select for. When they were evaluating a number of different criteria, they found that non-reproductive mites, infested brood removal, and the proportion of mites in the brood had very large heritable components, meaning they could select for those characteristics. What was really interesting is they only started this evaluation from only 28 colonies. So not really tremendously genetically diverse, but by having good selection criteria with a high heritability, they were able to select for bees that had what they found out was Varroa sensitive hygienic behavior. And in bees with Varroa sensitive hygienic behavior, or VSH, it works in this manner. So right before that honeybee larva turns into a pupa and that cell is capped, the Varroa will infest that cell. The Varroa then feeds on that larva as it transitions to a pupa and starts to lay eggs. And then in the white-eyed pupa stage, those eggs will hatch. And it's at this stage where the worker honeybees can detect that Varroa infestation. There's a chemical that they use that cues them into hey, this cell is infested. They open up that cell, and if they find out there's a Varroa in it, they remove that infested pupae and the associated with Varroa and throw it out of the colony. So this is very specific to Varroa. There's also general hygienic behavior, which gets rid of pupae that are infested with chalk brood or bacterial diseases. But Varroa sensitive hygienic behavior is specific to this Varroa infestation. That was the mechanism. So they got these bees that have low Varroa populations. Well, how do the colonies look? These colonies that have high levels of pure VSH don't really do so well. They kind of work like those colonies I mentioned that you find in nature. So they do have very low Varroa populations. You can see here 19 versus 834, so almost 20 times lower Varroa populations, which is good, right? You want low Varroa. You have very low Varroa in the brood cells, so about 2 orders of magnitude less. So again, nice when we're talking about Varroa, very desirable. However, the trade-off is that they have typically less capped brood. So this is just showing the number of cells per brood frame, and it's about half compared to the unselected one. So there's a lot less brood, and there's also smaller colony sizes, so fewer adults. So almost about 1/3 smaller colonies compared to the controls. Well, as beekeepers, you want lots of brood and you want lots of bees, right? So what they had to do was figure out a way to get over this, because there are downsides to every selective breeding. Because some other examples, besides what we just talked about with the VSH bees, is that like something like bananas that have been tremendously and intensively selected for these fruits that ship easy and high palatability, they're also susceptible to this Panama disease. It's kind of fungus, as you can see it here. So that's kind of a trade-off. Breeding large chickens for breast meat has this trade-off where they have these structural deficiencies that sometimes they have trouble walking. Same thing with breeding German Shepherds. This sort of selection for desirable characteristics causes a lot of problems, such as hip dysplasia, elbow dysplasia, and a myriad of other characters. So regardless of whether we're talking about plants or animals, intense breeding for these selective traits can have consequences, and bees are no different. So we wanted to have this low Varroa population bees, but we needed them to be better at producing honey, large colony sizes, lots of brood. So that's when we developed this POL-Line strain of honeybees. POL-Line just stands for pollination line, and you'll see why that is in a little bit. So POL-Line has high levels of Varroa sensitive hygienic behavior, or VSH, so they're good at controlling Varroa, but they've been outcrossed to commercial beekeepers for desirable traits such as rapid population growth, high honey production, high overwintering survivorship. And actually what's really important for me is gentleness. These bees are so easy to work. You just need a t-shirt and a veil and you can work them. You don't have to worry about them getting too upset and defensive. But they've been maintained by rigorous selection and propagated by instrumental insemination. So when we're breeding these queens, we have to physically inseminate them, kind of like how you would inseminate cattle. We actually do that with these bees, where we take drone semen and inseminate them. What we wanted to do for this project was to see if we could do open breeding. And that's how this project works, is that this blue star here shows the location of the lab, and this yellow area here is the location of a cooperating farmer, right adjacent to our lab. All these red dots here indicate the areas where we raise these POL-Line bees. So we have a large population that we can do open breeding with to see if we can put them into commercial beekeeping operations. The way that we bred these POL-Line bees is like this. So we had that highly resistant stock that was had very low Varroa, but poor beekeeping characteristics. We then shared that with 3 different beekeepers and allowed it to breed with their populations, because these beekeepers have been raising their queens to produce colonies that are big and have lots of brood, things that are very good for beekeepers. We then took those bees that survived, we ran them through the whole commercial beekeeping operation, taken for pollination and out to California. Then when they come back, we took those bees that survived and then back crossed them to the highly resistant stock. So that way we can make sure that they're going to have things like low Varroa population. But hopefully by working with these other beekeepers, we could get their genetics for large populations and large brood. We then take it back to the start of this project, and we did this 3 times. So for 3 consecutive years, we outcrossed this with different beekeepers. So that way we get the best genetics for having low Varroa but large honeybee populations. That's how we created this Polline stock. So as you'll see, Polline is very, very good at keeping Varroa populations low and having high colony survivorship, as you'll see. There are some advantages, some comparable traits, but also some disadvantages to this stock. It's not the silver bullet. It does have high overwinter survival, as you see on the next few slides. It does have low virus levels, which are very good. You know, we want to keep viruses low, and there's no real treatment for viruses. There's no chemical you put on the colonies. So this, you'll see these virus levels and how that works. We could potentially reduce miticide use because they could keep Varroa populations low on their own. They might need minimal chemical intervention, which is good. They have, and for me as a scientist, they have reduced impact of Varroa viruses and miticides on experimental design, which means it's a very cleaner system. So that way we can study honeybees without these confounding variables. So for me as a scientist, this is really good. So some of the things that these bees are comparable to other commercial bees are colony size. So especially at almonds, that's where our commercial beekeepers make their money, is by having large populations on almonds. So that's very comparable to commercial stocks. They do brood up very quickly. Down here in Louisiana, they will start brooding up big time in January even. And so kind of one of the things that we selected for was that rapid brood buildup early in the year for almond pollination. They do produce comparable amounts of honey. I have an asterisk there because there's a caveat to that. One of the limitations of this stock is that honey production can be reduced. Again, caveats to that, and you'll see it in the data coming up. They have high Varroa sensitivity, meaning that even if Varroa gets around to about 4 Varroa per 100 bees, this stock can die. Whereas other stocks they're fine with higher levels of Varroa. And you'll see what this looks like with some data coming later. There's limited availability of this stock. We can't send you these bees. We work with commercial beekeepers to make the stock available. It has limited availability now, but we're hoping to increase that going forward. So you could just call up a queen producer and get these queens. There's rapid genetic dilution of this trait. So there's about 12 different genes involved with VSH behavior. Once that queen, the daughter, she mates with other bees that are not VSH drones. And about the second generation, they're okay. But the second time they outcross, this trait goes away. So it's very hard to maintain these genetics. One of the consequences of this selective breeding is that there is high pesticide sensitivity in Polline across the board. I've tested many different pesticides compared to many different stocks, and Polline is usually almost always the most sensitive to pesticides. And one of the interesting things is that it's a very genetically distinct population. So it might not be representative of what's going on out there. I don't have time to talk about it, but it's interesting how genetically distinct these Polline bees are. All right, so let's look at the data. What are the experiments that we did? So this was part of a large 3-year project that involved us here at the lab in Baton Rouge. So Bob Danka and Mike Simoni-Finstrom. You can see those two here were co-investigators on this. And then also at the Beltsville lab in Maryland, Judy Chen, who's a virologist, was involved. We also worked with Kristen Healy and Dan Swale at LSU on this project. They are toxicologists and physiologists. We had a lot of different goals for this grant, but the one that we're going to talk about today is to determine if Polline, the bees with Varroa sensitive hygienic behavior, can mitigate losses from Varroa and associated viruses in commercial operations. That's what we're going to focus on today. We'll measure a lot of other things and they're still ongoing, but this is what we want to focus on today. So we started this project in 2017 and ran it into 2018. I'll talk about this first year's worth of data. We love to do big experiments. Sometimes when I read scientific papers on honeybees, they say, we used 8 colonies per treatment group and all that. We like to do big things. We work with commercial beekeepers who can move these colonies around. So we started with 171 Polline colonies and 191 commercial colonies of just standard commercial honeybee stock. So we're talking almost 400 colonies were in this study. It's a huge, massive data set. We sampled these honeybee colonies at 6 different times of the year. We took the first observations when we established the colonies here in Louisiana and Mississippi in April. We then sampled them again in June when they were moved up to the Dakotas for honey production. We then sampled them again in September after the honey was harvested in the Dakotas. And then in December, we sampled them in 2 different migratory routes. Some of the bees went out to California, and some of those bees came back here to Mississippi. In February of the following year, we sampled these bees that were out in California for almond pollination, out in actual almond orchards, or we looked at the ones just up the road in Mississippi. So we're trying to see if almond pollination had any effect on the bees here. And then finally, we sampled them in Mississippi in April of the following year when they come back for splitting and buildup. So we sampled these 6 different times throughout the year. We took a lot of different colony measurements. These are just a few of them, but on each colony, we took an insane amount of data. So this is just some of it. So Varroa infestation viruses, queen status, honeybee population, so frames of bees, how much honey did they make, how much brood, among other things. But today we're going to focus on those first 2, Varroa infestation and virus levels. What we found was very interesting. I like when we have data where you don't even need statistics. So this is just showing that Polline colonies have higher survival rate. On the x-axis here, we have the dates of our sampling intervals. So this May was the start of the project, April was the end of the project the following year. We then looked at colony survivorship. So how many colonies survived at each date. And as you can see from May, June, and September sampling dates, there was very similar survivorship between Polline and commercial colonies. However, between September and December, the commercial stock dropped off quite dramatically. And finally, by the end of the experiment, only about 25% of those colonies were still surviving. Polline, the ones with the Varroa sensitive hygienic behavior, maintained about 60% survivorship at the end. So Polline had about twice the survivorship in this experiment compared to the commercial stock. All other things being equal in this experiment. So just genetics had a tremendous influence. Well, why did Polline survive at a higher rate? It has a lot to do with Varroa. So when we looked at in September, when we looked at Varroa levels, we found that Polline across the board had about just over 2 Varroa per 100 bees, whereas in the commercial stock, they had about 8 Varroa per 100 bees. And so that's how we come up with this Varroa infestation rate. We count the total number of Varroa and standardize it by the sample, by the number of bees in the sample. So you can see here, Polline has about 4 times less Varroa than the commercial stock, all right? Well, it turns out that Polline is highly sensitive to Varroa infestation. In the colonies that died in that survived in Polline, that made it through April, in September, they had under 2 Varroa per 100 bees. But all the Polline colonies that died had about 4 mites per 100 bees in September. However, the commercial stock did just fine with 4 Varroa per 100 bees. But in the commercial stock, when they had about 10 Varroa per 100 bees in September, they were dead. So you can see here, Polline is uniquely sensitive to Varroa infestation. They try to keep those populations very, very low. So we could then answer the question, how much does Varroa influence colony losses? So what this graph here shows is Polline and commercial stock. Then we have commercial losses on the left. The black bars show that the total number of colonies that were lost over the course of the experiment. So this is from May of 2017 through April of 2018. If we identify the number of colonies that were lost that have 4 Varroa per 100 bees in Polline, and 10 mites per 100 bees in the commercial stock, as per that previous slide, we can estimate how many of those colonies were lost due to high mite infestation. So when we do that comparison, we can see that most colonies are lost due to high Varroa populations within that stock. So 37 of the 54 Polline colonies that lost had high Varroa populations, whereas 118 out of 120 in the commercial stock died because of high Varroa. So you can see here, Varroa are a tremendous influence on colony losses. So let's look at the viruses. So I mentioned before in the introduction that Varroa transmits viruses. So let's look at the viruses that Varroa transmits. CPV is chronic bee paralysis virus. DWV is deformed wing virus version 1. VDV stands for Varroa destructor virus 1. These VDV and DWV are different versions of the same virus. Deformed wing virus A, deformed wing virus B, we call it now. And these are the major viruses transmitted by Varroa. On this axis here, we have the mean viral quantity. So how many copies of those viruses do we find in the bees? And as you can see here, whether we're talking about chronic bee paralysis virus or deformed wing virus, Polline has significantly less virus in every instance compared to the commercial stock. Well, why do they have lower viruses? They have lower viruses because they have lower Varroa. This graph here shows that September Varroa population and the number of copies of deformed wing virus. As you get more Varroa, you get more viruses. It's pretty simple as that. More Varroa, more viruses. Well, the other way you could get less virus is if there's any kind of viral tolerance in Polline. Perhaps that was a secondary selective trait. So are the low virus levels due to low Varroa infestation, or is it viral resistance? So we could test that at the lab. And the way that we do it is like this. I like this experimental design. It's really cool. So here's how it works. We get a white eye pupa, and you can see that here. This is the pupa. And then we inject it with this needle. You see that here? This needle delivers a dose of deformed wing virus, 10 to the 5th copy. So 10,000 copies of deformed wing virus gets injected. We then raise that pupa and then evaluate the wing phenotype upon emergence. Because it's pretty easy to score. Deformed wing virus, you get those short curly wings. And what we did is we came up with 4 different classes: 0, 1, 2, and 3. If they score them as 0, the wings are normal. They look like there's no virus in them. If they have some slight malformation, so a little bit shorter wings, we call it slight. Moderate ones mean the wings are getting twisted. And then in severe cases, they're just reduced to those small little nubs. So when we did this and compared Polline to regular Italian stock, we found that Polline is no more resistant to deformed wing virus than unselected Italian stock. So Polline does not have virus resistance. It has Varroa resistance that keeps virus levels low. So by controlling the Varroa, you can control the virus in Polline stock. So as you can see here, they still get severe wing deformation when injected with deformed wing virus. So you can see here, the key to this whole equation is Varroa, 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 Varroa. If you take anything home, Varroa, Varroa, Varroa. We can also look at the honey production in these colonies, and I mentioned it was comparable. So in the first year in this study, compared to the commercial stock, there was not much of a difference. There was no statistical significant difference between commercial honey production and Polline honey production. It's a little less, but not significantly less. And you'll see the caveat here coming up in a little bit. So take-home messages from this first part is that high Varroa infestation is the major cause of colony losses. I hope that was abundantly clear with all that data that we showed. Bees selected for VSH behavior have reduced colony losses under migratory beekeeping conditions. And this is something that is universal for this stock, wherever we put it out. It tends to do very well, not just here in Louisiana, but the Dakotas, Maryland, Texas. So this is really good data. There is some stock differences in sensitivity to Varroa. And that's what I think is really interesting, because we have these thresholds for Varroa and they're usually conservative. However, I think we need to study each stock's own tolerance for Varroa to come up with stock specific treatment recommendations. And then the lower levels of Varroa transmitted viruses are due to less Varroa infestation and not virus resistance. So control Varroa, you have colonies that last longer and are healthier colonies. Well, part of what we do here at the USDA is to take this technology and get it into the beekeepers' hands. That is the ultimate goal of what we do. So translational research. And that's what's actually happening right now with this stock. Bob Danka is the former research leader at the lab who has since retired. And David Thomas is a beekeeper that has roots here in Louisiana, but also owns operations out in Hawaii. And they came up with the idea to take this stock of POL-Line bees with Varroa sensitive hygienic behavior and make them commercially available. So you just call them up, say, I want some of these bees, and they ship them to you. This is where they founded the Hilo Bee Company. HiloBees.com if you want to see anything more about it. So the Hilo Bee project the goal is to breed and distribute Varroa resistant bees. So this is the POL-Line bees that we sent them to Hawaii. They raise them, select them, and then raise them on large scale to make them commercially available. This is kind of tricky because you have to make sure that you have a homogeneous population, drone saturation areas. And so it's quite an involved operation. So they want to make these commercially available, hopefully soon. So we can do all the breeding we want and selection on the island, but we have to see how these bees actually perform in real world situations. So similar to what we did with those POL-Line bees, they're going to run them with these Hilo bees. So again, install them in May, evaluate the colonies in August and October, and then finally in February during almond production. So this is a currently ongoing project just to make sure that these bees are performing not only well on the island where they're being selected, but also in honeybee keeping operations. So what they found was actually pretty encouraging, is that the mite resistance is very good. So when the project started, the Hilo bees, these derived POL-Lines that are bred out in Hawaii, they had very low Varroa infestation in May. In August, they had significantly less Varroa infestation compared to the commercial Italian stock. What was interesting as part of this experiment is that miticides were only applied to the Italian stock. So Hilo did not get any Varroa miticide treatments, but in October, the levels of Varroa were nearly the same as they were in the Italian compared to the Hilo bees. The Hilo bees got no miticides. The Italians did get a miticide. And so what this is showing us is that this Varroa sensitive hygienic behavior is as good as a miticide treatment. That's pretty impressive. And then finally, in February, the numbers of Varroa come crashing down again. So Hilo bees with Varroa sensitive hygienic behavior do very well without chemical intervention. That's actually very, very important. They have enhanced overwinter survivorship and stronger colonies for almond pollination. And so when we look at the colony survival rate between Hilo and the Italian stocks in August, they're pretty comparable, 65 or 69% in Hilo and Italian respectively. But in February, in almonds, 53% of Hilo colonies survived compared to only 38% of the commercial Italian stock. While it is a pretty substantial difference, this is actually pretty important for beekeepers because every colony that goes out to almonds in February is worth at least $200 in revenue. So you can see here, having more colonies going out to almonds is extremely important. However, again, the caveat to honey production really showed up here with these Hilo bees. So the Hilo bees that have Varroa sensitive hygienic behavior produced about 25% less honey in this situation. So yeah, it does matter where these bees are kept and what stock of bees, but Varroa sensitive hygiene tends to have a production cost of less honey. The other caveat to this is also under drought conditions, these VSH bees don't perform super well. They don't make a lot of honey, and they also die off at a pretty decent rate because of they apparently are not good at dealing with stressful environments such as drought. So again, some caveats. All right, so let's finish up here about the Hilo bee breeding project. Is that they're going to continue breeding and refining this population to improve productivity. They want to make that honey production comparable, so that's going to take some breeding. Keep field testing it to get it to where the bees are ready for distribution. These are not going to be released just because people want them. They're going to be released when the bees are ready, when they're performing well in these operations. So the take-home messages I want you to have for tonight is that behavioral resistance to Varroa is a viable option. This isn't something that we create at the lab that only works here. This is a viable option in commercial beekeeping operations and even backyard beekeeping operations. If you want to go chemical-free, this behavioral resistance or VSH resistance is probably one of the best tools you can have in your toolbox. It's also really important to utilize multiple Varroa control methods to ensure that you have redundancy in your Varroa control program and also to efficiency. We mentioned about how these VSH bees, the Hilo bees, they can provide Varroa control just as good as a miticide treatment. However, having a miticide also makes sure that these Varroa are going to get knocked down. Don't rely just on miticides or just on VSH bees, but you have a complementation between those and also other methods too, such as brood breaks and things like that. We can have better bees through superior genetics. We're always working on improving our stocks and making them available to beekeepers, but also to, if you can select from your own bees, the ones that have the most desirable characteristics, something that you're looking for in your operation that you want, that's really important. So this breeding is actually pretty simple, but you have to do it on a large scale. So VSH queens are important for all beekeepers, but ask questions of vendors. We have been seeing with all this VSH behavior that's been coming out, we have been seeing people marketing VSH queens. However, it's really important to know how they're selecting for this trait, when the last time they evaluated their bees, where they got their original material. Did they get it from us at the USDA? Did they get it from somewhere else? Ask hard questions because a lot of them can't answer that. They are very good at what they do. However, I do see people all of a sudden saying, oh, we have VSH queens, and they don't say where they got their bees from, where they got their stock. So do yourself a favor and ask them lots of questions. The good vendors will have the right answers. So I didn't do all this work myself. I've actually worked with a lot of great people at the USDA Honeybee Lab. So Bob Dank and Mike Simone Finstrom were the major collaborators, and we have a great group of technicians that travel on the road and work with us in the field. LSU has been very important too. So the two professors, Kristen and Daniel, then a number of graduate students and postdocs, but most importantly, our industry partners that move our bees around and help us out, because without them, we can't move 400 colonies around very easily. Then with the Hilo Bee Project, the Hawaii Island Honey Company with Dave Thomas, Arista Bee Research with Bart Jan Verhoet, who does the breeding programs. Zach Browning of Browning Honey Company was doing field trials for us up in the Dakotas. And Project Apis M and Daniel Downey were major funders for the Hilo Bee Project, as well as a USDA grant. So with that, I'm very happy to take any questions that you might have. And I have nowhere else to be, so I can stay here as long as you need me to. So let's have a chat.
Good questions. I've got a few.
Okay, let's go.
So is viral resistance a trait that's known to already exist?
So does viral resistance exist? Yeah, there is a couple different ways. So there's two different things that bees could do to withstand viruses. One is they could have their immune system break down the viruses and kill them. There's a kind of immunity that exists that way. Another way they could do it is what's called viral tolerance, where they could have high levels of viruses, but they don't die from it. There's a professor at University of Minnesota who's very into that idea, and it's called super competitive exclusion, where it just basically says that they could have all these viruses, but the receptors are in a way that it doesn't kill them. So we have identified that in certain honeybee stocks. So there is a stock of bees from Gotland is in the Netherlands. Is Gotland? I forget where Gotland is. It's over in Europe somewhere. I'm drawing a blank. But those bees have this high viral tolerance. We've tried to select for viral resistance, so killing the virus. The way that we do that is you inject it with the known amount of virus and see if that virus level goes down. It's really difficult because there seems to be a large paternal effect with that behavior, and it really doesn't get the virus levels down to where we need them, even after about 4 rounds of breeding for it. It seems that there might be way too many genes involved to get us at that, but it's still an ongoing project at the lab for sure.
Okay, yeah, that was my follow-up question was, is it heritable, and is anybody trying?
Yes. Yeah,
anyway.
because we know the genes involved with viral resistance. So it's just a matter of getting them in the right frequencies with all the other sorts of genes that they need to withstand viruses, because it's the immune system itself that's really responsible for it. But there's all these other biological pathways that they need to amp up to get viral resistance. It's just that we might not have found the appropriate combination yet. But we're going to keep trying, because viral resistance would be great. There's nothing you can really do for viruses at the colony level except keep your Varroa low. So there's no kind of chemical treatment. Yeah. Also, wait, one more thing to follow up on that. We've actually irradiated honeybees, like how a lot of our food is irradiated. So we hit them with really high levels of radiation, and we can kill some viruses, but when we inject those into bees, they're still bad. So there's nothing really can really do too much about viruses except keep Varroa levels low.
Okay. And I know this isn't a question for you theoretically, but this is always when we hear about breeding programs like this that are going to sell into commercial operations, and they want to sell commercial quantities of queens. Mm-hmm. In your experience, what is the commercial quantity? Like, how big of an order would we have to gather throughout all of central Pennsylvania when if these are available, right?
Yeah, so that's the interesting thing, is that demand for queens across the queen producing industry is way higher than demand. I'm sure everyone who's ordered queens know you have to order them first thing in January, or you might not get any. But for the Hilo Bee project, what they really want to do is make over 100,000 of these queens commercially available. Now, the way that they get at that is through the Hilo company, but they're starting to maybe partner with other people to kind of push this out and grow this VSH availability. The idea is to make it available enough where we can get it into the populations that they then sustain themselves. Like I mentioned before, this VSH trait goes away if those queens cross to non-VSH drones, and that happens very quickly. If we could get most of the bees to be these VSH bees, the probability of their crossing to a VSH drone goes up. And I mentioned we could do that. We've done that here at the lab. We open mate these queens and they're fine. We get a few stinkers here and there, but a great proportion of them are open mated queens that do well. So yeah, they really want to ramp up this operation. But the biggest thing that they've been doing the first few years here is making sure that the breeding and genetics are airtight before they start selling these things. Because what happened was that original stock of VSH was made available to the beekeeping industry, but people didn't like it so much because they had small colony sizes and they had very little brood. Two of the biggest things that are attractive to beekeepers. So it kind of got a little bit of a bad rap early on. So that's why they're putting a lot of effort into making sure that these are the best bees they could be.
Yeah, go ahead. I got a question.
Yep.
Did you make a statement earlier that the VHS bees were more sensitive to pesticides?
Yeah, that's true. That's true.
Which pesticides?
So I I have my big three. So one is a pyrethroid insecticide called phenothryn that's used a lot in mosquito control operations. We then have an organophosphate called chlorpyrifos, which is another organophosphate used a lot in agriculture, especially soybean aphids, for example. It's found everywhere and it's really it's cheap. That's why people use it a lot. And then we also look at chlorphenidin, which is a neonicotinoid. So we cover the three major classes of insecticides. And whenever we do stock evaluations, Polline almost always comes out as the most sensitive. We published this paper maybe 2 years ago now, where we looked at bees of different genetic diversity. We looked at these highly inbred Polline bees that have been very selectively bred. And then we compared them to other bees of varying genetic heterogeneity. So we looked at ones that were commercially kept, ones that were open, and then feral bees. And what we found is as you increase your selective events, selection criteria, it tends to make them more sensitive to pesticides. And to me, that just makes a lot of sense because like I mentioned that there are trade-offs to selective breeding. And it could be potentially that this pesticide sensitivity is one of those trade-offs. And when I say sensitive, I don't mean like a little drop will affect them. It's a statistically significant increase in sensitivity, but it's not this thing where if they see any pesticide, it's going to kill them. That study I mentioned, we only found about a 3-fold difference between the most tolerant and least tolerant in Polline. When we look at the adults, it might only be a few fold difference. It's not a 10,000 fold difference between stocks, but it is something that we do have to pay attention to.
Anyone else?
Because,
Questions?
well, just to follow up on that is part of the thing that we looked at in this big experiment was pesticide exposure on honeybees and how that affects them. So we take pollen, send it to a lab, they tell us what the levels are, what the levels of those pesticides are in the pollen. And we didn't see any difference in survivorship between Polline and the commercial stock. So that tells us that, yeah, that we might be able to find that in the lab, but in the field, it might not matter so much.
How long are your queens lasting? Are we testing for that? Or?
Yeah, so that's a good point. So queen longevity is actually one of the things that we do breed from. So Garrett is our queen breeder, and he likes to breed from old queens because the idea is that old queens, if you breed from them, they should have characteristics that should lead to old queens. We have 3-year-old Polline queens here at the lab. That's here at the lab where they're very well taken care of. But in commercial beekeeping operations, we do find that these queens supersede less compared to commercial bees. And we haven't really looked at longevity in commercial beekeeping operations, mostly because some of the beekeepers that we work with, they're requeening every 6 months or every year anyway. So, but we haven't done that. But I know here at the lab, we do have queens that are 3 years old.
I had a quick question. Has your research shown any adaptability of the Varroa mites to any other type of pollinators, like any other type of bees, that could sort of then come around back again to the honeybees?
Yeah, so short answer, no, they don't really infest any other bees besides honeybees and their native host, the Apis cerana, the Easter honeybee. So Varroa evolved with Apis cerana over in like Southeast Asia, and they kind of live in this sort of harmony where, yeah, the parasites will eat some of their brood, but they also tolerate it. The bees have ways of dealing with it. So it's kind of this equilibrium. They've had a long time to figure that out. But when these Varroa moved onto honeybees in the early 1900s, honeybees were completely naive to them. So they were a good host for it without any defenses. So that's why it spread through them. As far as other bees go, you could maybe find coincidental infestations of Varroa, but they don't reproduce on them. That's the major thing. Because One of the things that Varroa do is they feed on this very specific part of the honeybee called the fat body. And a fat body is basically an insect's liver. It functions very similar to ours. And that's what they need to survive. They need certain all these certain nutrients from it. Other bees, like bumblebees or solitary bees, they don't have that sort of nutrition of their fat body. They are really co-evolved with honeybees, or specifically bees in the Apis, in the genus Apis. So Apis mellifera, Apis cerana, and things like that. They also need a lot of brood to reproduce on. So honeybees, Apis cerana, our bees, the eastern honeybees, Apis cerana, they have pretty large colony sizes, so there's ample opportunity. A lot of other bees are solitary or semi-social, and they have very small colony sizes, so they tend not to do well in those environments. Yeah, a lot of it too. Also, it comes down to timing. So the Varroa's lifespan is synchronized with the honeybee and Apis cerana, the eastern honeybee, their brood cycles. So it has to be a very specific set of durations as well.
I've got another question. It's sort of off topic, but you started drifting off topic. So
Okay. Yeah. I can answer any questions you have.
it was like maybe 4 or 5 years ago that the researchers that demonstrated that the Varroa were feeding on the fat bodies.
Yep.
What was the guy's name? Sam
Ramsey.
Sam Ramsey. Yep.
So the expectation was that with this revelation that they're feeding on the fat bodies, there would be new scientific ways introduced to control Varroa based on them feeding on the fat bodies. I haven't seen anything.
Yeah. So the difficult part of doing this research with Varroa, especially the reproductive and feeding part, is that we don't have a good way to do this in the lab. We usually have to wait to midsummer through early fall to have enough Varroa to do any of these experiments. So that makes it pretty difficult. I know that they've been having a challenge of actually translating. So basically what they wanted to do was be able to feed the bees something that gets into that fat body, but then goes out and kills a Varroa. So they had some promising leads early, and they took it out of the lab and into the field. But when they took it to the field, it didn't translate. So that's kind of a big thing in honeybee research is, yeah, it works in the lab, but not necessarily at the field. The other thing that happened was the materials that they were able to give to the honeybees in the lab actually turned out to be pretty toxic in the field. So that happens often. Because I don't know if you remember, but there was a couple years ago, there was this idea that you could feed bees lithium chloride and that will kill Varroa. I don't know if you heard about this one, but it was kind of this serendipitous experiment. And in the lab, oh yeah, this works great and it's fantastic. And we could kill Varroa like this if we feed the adult bees this lithium chloride in the lab. Great. It turns out lithium chloride is really toxic to honeybee larvae. So it's things like that that sound promising, but sometimes don't translate. Yeah.
The operation was a success, but the patient died.
Excuse me?
The operation was a success, but the patient died.
Yes, that kind of stuff definitely happens. Yeah, and that's kind of the frustrating thing with even like doing miticide research. So we're trying to find other miticides besides amitraz, formic acid, or all these other things. But again, it's hard to find something because what we do is we say, does it kill Varroa? Yes or no? Yes, proceed to step 2. Does it kill honeybees? Yes or no? Doesn't kill honeybees? Okay, proceed to step 3. How does it work in the colony? Because getting it onto the Varroa in the colony, it's a very, very different system than in the lab. So we've had a number of different things that showed promise that just don't make it there. It's like drug discovery. There's a lot of things that look like they're going to fight cancer, but turns out can't get them into patients.
You mentioned in the almond fields.
Yep.
Why do you put the your bees with the commercial bees together?
Yes, they are in the same apiaries.
Okay, how can you graft that because of mixture then? When
I'm
they-
sorry, what,
When they're
I'm
mixed
sorry,
together, when they're mixed together, how can you graft that then?
so the colonies are in the same apiary, and
Right.
at that point, yeah, we're not. Are you asking about?
The mixture of both bees together.
Yeah, so actually even before almonds, and when they're making honey up in the Dakotas, they're in the same apiary side by side. And so that actually allows us to compare how that landscape is affecting. So we want to keep that landscape equal, right? Because landscape is very, very important to honeybees. So when we take them up to the Dakotas, yeah, they're all in the same apiaries together. So are you asking how, what are you thinking about? What aspect are the mixing? Is it just the competition for floral resources?
Well, bee drift, yeah.
Oh yeah,
Yeah.
yeah. Bee drift definitely happens. Because in almonds in California, you're getting what 2 colonies per acre, and they stack them up pretty. They're right next to each other. So yeah, drift definitely occurs. But we didn't really measure that in this experiment. We tried when we were out there trying to look at the colony sizes. We try to get out there early in the morning as possible, usually just before daybreak, so that way they have the maximum number of foragers. That's kind of one of the ways that we tried to minimize any of those observations of drifting. But yeah, we haven't. I'm sure it happens, but we haven't quantified that. Good question though.
Anyone else? Anyone else online?
Keep asking. I like questions.
No? All right. Well. 3, 2, 1. Okay. You had your chance.
Good.
Great. Yeah. So thank you so much, Frank. Yeah. This is great. This is really interesting.
Well, good.
I'm so glad that you were able to do it. Glad we in Zoom were able to bring it to you guys.
Yeah. After I got it up and running, yeah, good.
Yeah. So for those on the call, we now have a YouTube channel because I didn't know how else to do it, and it seemed like a good idea. The Mike Palmer talk is posted there. This talk will be posted shortly. So I just gotta let the computer crank through it and get it uploaded.
What's YouTube channel called?
I hope that if you search for Lancaster County Beekeeper Society, that it would find it. Yeah, I had made a Google account. Google owns YouTube, right? So they're all interconnected. I had made one with specifically for the Bee Club, a Google account a couple of years ago. That's how we do the mapping thing with the swarm list. Unfortunately, they have my old cell phone number, which I no longer have. And once that happens, you are skunked. Tough crap for you. You make a new account. Once they can't text that number anymore. So I had to make a new account. So just the trials and tribulations of dealing with technology companies. So anyway, great. Thank you very much for coming. Thank you again, Dr. Rinkevich, for your time. And
My pleasure. My
your
pleasure.
awesome slides. And thank you for letting us record it, and we will get it posted. And yeah, so upcoming stuff. May's a weird one, right? Meetings next week already, right? May 17th. It's because it's one of those weird ones that it feels early. So May 17th, next Tuesday at the farm on Auction Road. 6 o'clock will be our meeting. And we're also the following week, because May has 5 weeks and we did kind of wanted to shimmy this around. The following week on Thursday, we will have a hive check. So for anybody on the call who can't ever make a Tuesday thing, right? That's one of the reasons we're doing these meetings, right? Because these aren't Tuesdays. Who can't ever make a Tuesday thing, that is the 26th, I believe. These dates are on the website. Yes, that'll be the 26th. And I did check in with Alyssa at the farm, and we're not stepping on any issues with the planting trials that go on out there for our dates. Yeah, so great, thanks again. And okay, great. I'll end the meeting and we'll get this posted as soon as we can. Thanks again.
All right, folks. Happy beekeeping. Bye-bye.